Hello, Miss Gina and all my friends. Today, me and my group will present about the paper with title "Physical Basis for the Loading of a Bacterial Replicative Helicase onto DNA." So, this is the name of my group: Chetishta, Jingga, Nicholas, Silvan Saputra, and Willie Elsan. Introduction. DNA replication is very important in all living cells because in order to proliferate, they must first replicate their DNA. And to replicate DNA, they use helicase to unwind the double helix form of DNA. However, the DNA bubble is topologically inaccessible to helicase because helicase has a hexameric closed ring system. And the question from this paper is how helicase access the DNA. So this is the component of helicase. It comprises of DNA B, the main unit of helicase, and DNA C, the loading factor. And when they join together, they form the DNA B-C complexes.
I will continue the presentation. Two forms of helicase. Two forms of DNA B is oscillating. They continuously change its conformation into DNA B dilated and DNA B constricted form when it's moving along the strand to unwind the DNA double helix. In particular, NTD or N-terminal domain will undergo a large conformational rearrangement. It will transition from a widened or elliptical dilated form encircled a broad central channel into a highly constricted form into a narrow port, or it is called constricted form. The aim of the research is to uncover the underlying mechanism about how bacterial helicase being loaded onto the DNA strand prior the replication process in. Research method. The methods of DNAB and DNAC observation. The DNAB and DNAC is obtained from genetically engineered E. coli that can overexpress these proteins. The DNAB and DNAC is isolated and purified based on their molecular size, separated by using gel chromatography column. It creating DNAC ssDNA assay and loaded into the purified DNAB solution, and it is creating the ATP hydrolysis assay to assess the ATP in the helicase loading. The high-resolution cryogenic electron microscopy is used for the observation. The image processing by using computer and protein model building.
Next, we're going to talk about the result and discussion. Now, on the topic of the structure of DNA-BC complex, it consists of 6 DNA-B and 6 DNA-C subunits in the dodecametric shape with a crack on the particle for the ssDNA binding. Now, it consists of 3 domains, the lower, the middle, and the upper domain, while the upper domain and the middle domain are joined by the linker helix, except the uppermost region of the upper domain where a crack is present. Next is the DNA-B recognition by DNA-C. Now, in high-resolution cryo-EM model, it's shown that the N-terminal region of the DNA-C alters the helix linker protein, leading to conformational changes in the helicase. Now, the details of the process can be seen in the next bullet. Next is the ATP turnover by DNA-C. Now, the nucleotide binding site of DNA-C occupy the ATP-like configuration, while the DNA-B occupy the ADP-like configuration. Now, this creates a hypothesis that the ATPase activity of DNA-C only occur when both ssDNA and the DNA-B is present. Now, to test this, in a catalytically inactive Walker A mutant of DNA-C showed that the ATPase activity only occur in the background level, confirming that DNA-C is responsible for the observed turnover of nucleotide. In conclusion, DNA-C requires ATP to do its conformational change to constrict the helicase after it is loaded into the ssDNA, binding the helicase into the DNA.
Okay, now I'm going to explain about the mechanism of ssDNA binding by DNA-C. The DNA-C promoter will occupy the ATP-bound configuration, where its subunit will disengage from its active site, causing conformation of the 6 loaded subunits to stick to the narrow cracked ring hexamer that encircle the nucleic acid substrates. The loop connecting each DNA-C ATPase domain to its corresponding N-terminal extension also undergoes a large, more than 90 degrees outward rotation. And 5 of the 6 DNA-C ATPase domain is engaged to each ssDNA. The nucleic acid is most kinked and less well ordered at the junction between DNA-C and DNA-B. This mechanism will produce the residue of phenylalanine 146, serine 177, and tyrosine 179. Analysis showed that the loss of any of these three residues in the DNA-C will lead to the defect in helicases loading. So what is the effect of nsDNA binding? First, the ATPase modules of DNA-B will tilt downward by 35 degrees, creating less spiral conformation. The nucleotide exchange during domain reorganization and 60 degrees rotation of DNA-B RICA, followed by their associated entities, forming dilated configuration. And last, the chains require one of the terminal subunit in the split ring hexamer to detach from one partner and re-engage with another. In return, it will allow the linker helix of the transitioning subunit to topologically seal the break in the split hexameric ring.
And now, the conclusion. There are 3 points in helicase opening or closing mechanism from this presentation. The first one is the DNA-C promotes constricted form of DNA-BC complex via ATP turnover. Second one, the remodeling of the interdomain and inter-subunit interaction in DNA-B will result in disengagement of the linker helix. The last one, the form pathway for helicase will be loaded onto the ssDNA. And that's all for our presentation. Thank you so much for your attention and have a great day.